Welcome friends. Today our topic is general characters of gymnosperms. The word gymnosperm has been derived from a Greek word where gymnos means naked and sperma means seed. Thus, the gymnosperms are a group of plants with naked seeds. Gymnosperms originated during the late Paleozoic era but flourished well during Mesozoic era. Hence, the Mesozoic era is called the age of gymnosperms. Some gymnosperms like Ginko biloba, Cycas, Metasquoia are called living fossils, as these plants are found both in living and fossil forms. There are about 70 genera with 725 living species of gymnosperms in the world today. Occurrence and distribution. Gymnosperms are found both in tropical and temperate regions. Some are found in Arctic regions. Of these, majority belong to conifers. The conifers are mostly distributed in the cooler regions of Europe, Asia and North America. They are dominant in upper timberline zone of mountain range, including the Himalayas. The living gymnosperms are widely distributed in the cold climates where snow rather than rain is the source of water. The only native gymnosperms which thrive in warm areas are the Cycas. In India, mostly the conifers thrive well in hilly areas. Very few gymnosperms except some Cycas grow in plains. Araucaria is grown as an ornamental plant in gardens. Now habit. Living gymnosperms are mostly perennials, xerophytic, evergreen and woody plants. Gymnosperms vary in their size. Sequoia sempervirens, a gymnosperm, is the tallest living plant in the world, reaching a height of about 125 meters and girth of about 30 meters. It is also known as coast redwood of California. External morphology. The plant body of gymnosperm is sporophyte and is differentiated into root, stem and leaves. Plants possess well developed taproot system. Root often shows symbiotic association with algae or with fungi. In Cycas, carotoid roots are present in addition to normal roots. The stem is aerial, erect, woody, unbranched as in Cycas or branched as in citrus. In some genera like Pinus, the branches are of two types. Number 1, long shoots or branches of unlimited growth, and number 2 is dwarf shoots or branches of limited growth. The leaves are mostly xerophytic, evergreen and simple or compound. They vary in size from small scaly leaf to more than 6 feet long leaves as in Cycas. The leaves of pines are of two types, scale leaves and foliage leaves. Scale leaves are minute, membranous, brown and deciduous. Foliage leaves are present on dwarf shoots only and are known as foliage spurs. They are green, simple, needle-like or pinnately compound. The leaves of Cycas exhibit circinate vernation, a characteristic feature of ferns. The arrangement of leaves may be opposed, and decussate as in netum, whorled as in sudrus, or spirally arranged as in texas. Now, the internal structure starting with the stem. Anatomically, the stem shows an epidermis, cortex and stele. The epidermis is single layered and very soon replaced by periderm. Cortex is parenchymatous. Based on the relative quantity of soft tissue and mechanical tissue, gymnosperms are classified into monozylic and pycnozylic types. In monozylic forms, as in Cycas, the wood is not compact and has parenchyma. In pycnozylic forms, wood is compact and relatively less amount of soft tissue is present. The stele is surrounded by a typical epidermis followed by a layer of pericycle. The vascular system consists of a number of vascular bundles arranged in the form of a ring. The vascular bundles are collateral, endog and open. This type of stele is called eustele. Phloem consists of only sieve tubes and phloem parenchyma. Companion cells are absent. Xylem consists of tracheids and xylem parenchyma only. Vessels are absent except in netails. The xylem is either endog or mesoarch. Secondary growth is seen in all the members. It is usually normal. However, anomalous secondary growth may be found in Cycas and netum. Normal secondary growth takes place due to the activity of fascicular cambium, which produces the typical annual rings. Internal structure of leaf. Anatomically, the leaf exhibits xerophytic characters. The epidermis is always covered by a cuticle. The stomata are sunken. The mesophyll may be differentiated into palisade and spongy parenchyma as in Cycas, or differentiated with arm parenchyma as in Pinus. In some members as in Cycas, with a single veined leaf, a transfusion tissue is present to help in lateral conduction, necessitated by the absence of side veins. The stomata are sunken and often present on pits. The stomata are either syndactylic or haplochilic. In syndactylic type, guard cell and subsidiary cells originated from the same mother cell. In haplochilic type, the guard cells and subsidiary cells originate from the different mother cells. In netum, both types of stomata can be seen. Stomata may be distributed on both sides of the leaf as in Pinus and Ginko, or confined to lower side as in Cycas. Now, the internal structure of root. Most of the gymnosperm roots show a typical dicotyledonous root structure. The xylem is either diarch or triarch. Cortex is mostly parenchymatous. Secondary growth is seen both in the cortex and in the stele. Now, the reproduction. Vegetative reproduction is very rare and is seen in some cycads where it is brought about by the formation of bulbils. Spore formation is the typical mode of reproduction in gymnosperms. All the gymnosperms are heterospores, that is, they produce two different kinds of spores, the male microspores and the female megaspores. The spores are born inside the sporangia. The two types of sporangia are born on special leaf-like structure called sporophylls. The microsporangia or pollen sacs are born on microsporophylls or stamens, and the megasporangia or ovules are borne on megasporophylls or carpels. The sporophylls are usually aggregated in the form of compact structures called cones or strobili. Plants may be monoecious or dioecious with referenced strobili. Very rarely, bisporangiate strobili may be found. Male cones are generally short-lived and smaller than female cones. Microsporophylls may be flat as in Cycas or peltate as in Pinus. Female cones are usually long-lived. They sometimes remain on the plant for several years till maturity and ripening of the seeds. Megasporophylls may be foliar or leaf-like and not aggregated in the form of cones as in Cycas, or colline and aggregated in the form of cones as in Pinus. Microsporangia are borne on the lower surface of microsporophylls. They may be numerous and grouped into sori as in Cycas or reduced to two as in Pinus. In Cycas, the male cones are the largest among the gymnosperms and arise singly at the top of the plant. Female cones are not formed, but megasporophylls are loosely arranged around the stem apex. In Pinus, the male cones arise in clusters at the end of the long shoots, in the axil of scale leaves. Female cones are large and are arranged in 2 or 3 pairs on the long shoots. In Netum, the reproductive structures are known as inflorescence. The male inflorescence or male cone consists a long axis on which cup-shaped structures called cupules are formed by the fusion of bracts. In the axil of these cups, male flowers are born. The female inflorescence or female cone also contains a long axis with cupules. In the axil of these cupules, 4 to 10 female flowers or ovules are arranged in a ring. Bisporangiate strobili are also noticed in some species of Netum. Now, ovule and female gametophyte. In Gymnosperms, ovules are naked and borne on megasporophylls. The ovule consists of central mass of parenchymatous tissue called nucellus. The nucellus is surrounded by integuments which covers the whole nucellus except at the top, where it leaves a small passage called micropyle. The integument is differentiated into an outer fleshy layer, middle stony layer and the inner fleshy layer. The apex of the nucellus develops into pollen chamber, where pollen grains undergo a period of rest after pollination. The development of female gametophyte begins with the differentiation of megaspore mother cell in the nucellus. Its development is either monosporic or tetrasporic. Megaspore mother cell undergoes meiosis to form linear tetrad of 4 megaspores, out of which the upper 3 degenerate and the lowermost remains as functional megaspore. The functional megaspore then develops into female gametophyte. Archegonia are formed towards the micropylar end of the female gametophyte. In Netum, archegonia are not formed. A few nuclei at the micropylar end of the female gametophyte act as egg, which is an angiospermic character. The archegonia of Gymnosperms do not have neck canal cells and ventral canal cells. Now the microspore and male gametophyte. Pollen grains or microspores are unicellular. In Pinus, pollen grains are winged. Microspores develop into male gametophyte. During the germination of microspores, one or two prothelial cells are cut off at one end, leaving the large antheridial cell which divides into generative cell and larger tube cell. The generative cell divides into stalk cell and body cell. Body cell produces two male sperms. The tube cell forms the pollen tube which acts as carrier of sperms. Now the pollination. In Gymnosperms, pollination is anemophilous. The ovule secretes mucilaginous substance which oozes out from the micropyle of the ovule and forms a pollination drop which traps the pollen grains. The pollen grains are drawn on due to the drying of pollination drop. Further changes take place in the pollen chamber. The pollen tube grows through the tissue of the nucellus until it reaches the neck of the archegonia. Now the fertilization. Gymnosperms do not require water for fertilization. At the time of fertilization, the tip of the pollen tube bursts and releases the sperms. The sperms swim to the neck of the archegonia with the help of cilia, and one of the sperms fuses with the egg to form the zygote. In Pinus and Netum, the sperms are non-motile, which is an advanced character. This type of fertilization is called siphonogamy. Now the young sporophyte. After fertilization, the zygote is formed. In Gymnosperms, the development of zygote is meroblastic, in which only the embryonal part develops into an embryo or embryos, while the hysterial and suspensorial part do not take part in the formation of embryo. Embryo is straight with exception in benign tails. Now the embryogeny. In Gymnosperms, except in Netum, the diploid nucleus undergoes free nuclear division. In Cycas, as many as 1000 or more nuclei are formed as a result of free nuclear division. In Pinus, only 4 nuclei are formed. After that, the wall formation begins, resulting in the formation of 16-celled proembryo. Now the polyembryony. Polyembryony is the characteristic feature of Gymnosperms. In Cycas, more than one archegonia are fertilized, resulting in the formation of more than one embryo. This phenomenon is known as simple polyembryony. In Conifers, polyembryony is due to the separation of 4 embryonal cells, and each of them develops into an embryo. This phenomenon is called cleavage polyembryony. It occurs in Pinus, but only one embryo attains maturity and the rest degenerate. In Gymnosperms, the embryo is differentiated into root, stem and leaf. It is endoscopic, which means plumule is directed away from the micropyle and radical towards the micropyle. Now the seed. After fertilization, the ovule develops into seed. The inner fleshy layer of ovule turns into tegmen, and middle stony layer forms testa or the seed coat, which protects inner embryo. Embryo is developed at the end of the long suspensor, which is pushed down into the food-laden endosperm They develops before fertilization. Female gametophyte, soon after it has absorbed the food from the nucellus, becomes endosperm. As there is no double fertilization or triple fusion, the endosperm is a haploid tissue. The whole of the nucellus except a small portion is crushed. The remaining small part of the nucellus persists in the seed in the form of a narrow film or cape known as perisperm. Since there is no closed ovary, true fruits like that of angiosperms are not found in gymnosperms. The developed ovules remain naked. Middle stony layer of the integument becomes extremely hard and protects the female gametophyte and embryo. Cotyledons may be one or two as in Cycas, or a whorl of many as in Pinus. They become green while still enclosed within the seeds. The seed generally undergoes a period of dormancy. However, the seed of Ginko and Cycas germinate without undergoing a period of rest. During germination, the seed coat ruptures, a radicle grows downwards and develops into primary taproot, while the plumule grows upright and develops stem whose growth is mostly unlimited except in Welwitschia. Sporophytic generation is diploid and is dominant and independent, while as gametophytic generation is much reduced and dependent. Now the classification of gymnosperms. Chamberlain in 1934 divided the gymnosperms into two large groups. Number 1, Cycadophyta. In this group, sporophylls are in cones. It includes three groups: Cycadophyllicales, Benititales and Cycadales. Number 2 is Coniferophyta. This group includes four orders: Ginkoales, Corditales, Coniferales and Nitales. Of these, Cycadophyllicales, Benititales and Corditales are totally extinct. Cycadales, Ginkoales, Coniferales and Nitales are living. So with this we have come to the end of this lecture that was about the general characters of gymnosperms. In this lecture we discussed morphology, reproduction, affinities and classification of gymnosperms in general. Thank you.